Hi everyone. So first of all, I'd like to thank Hikaru and Ivan and everyone from uh, SKCM² for inviting me. I'm really happy to be here today, and I'll be talking a little bit about our research, and afterwards I'm really happy to discuss if there's something we can work on together. So a little bit about myself. I have a very little experience in physics, so I studied chemistry and business in my undergrad. I studied proteins, I studied lipids, and then during my PhD, I studied RNA. And so, I studied all these biomolecules, proteins, lipids, RNA, and so that got me really interested. Why did life choose these biomolecules in particular, and how did they come to be in the first place? So that led me to my current institute, ELSI, where I've been since 2017, and I've been working on a lot of projects related to origins of life, astrobiology, kind of encompassing a lot of different techniques and fields. And so today, first of all, I'd like to start with a little simple discussion about what exactly is astrobiology and how does origins of life fit within this, because maybe this is a little bit outside of many of what you typically think of. And so in my opinion, of course, everyone's definition might be a little bit different. In my opinion, astrobiology incorporates a number of specific topics. One is the active search for life in space. For example, search for signs of current life, signs of former life in space, and this is possibly through, for example, missions to other planets, Cassini, Curiosity, or missions to moons or other planetary bodies like Hayabusa 2 or MMX. Another aspect is the active search for habitable planets in space. So, for example, search for exoplanets, probing the habitability of different extraterrestrial bodies, such as through giant telescopes like JWST or various radio telescopes. And also the active search for the origins of planets, Earth, and life on Earth. And specifically, this might involve, for example, search for organisms that lived in extreme environments that could have mimicked ancient Earth environments. Research on how Earth formed or various geological formations formed. And this might be through, for example, research on various analog environments on Earth that might correspond to an environment outside of Earth. And within this, there's also a few other topics, for example, the active search for how to make synthetic living systems in the lab, the active search of how primitive chemicals assembled into primitive life forms, and the active search of how primitive life evolved into modern life. I want to emphasize that it is active, and these definitions are certainly changing. And so origins of life, the research field origins of life, which is fairly broad in concept, kind of probably incorporates these three last topics, and this is what I've been working on in our lab. And it's actually also really important for us to be collaborating with a lot of researchers from different fields, because in order to answer these questions, you really have to understand so many different aspects of these primitive systems. So now, okay, let's take a step back. Let's focus on this. This is the cell. It's it's the basic unit of modern life. Most people believe that. If you ask some virus people, they might believe differently. But let's assume that the cell is the basic unit of modern life. And cell is very complex. It includes a lot of moving parts, moving reactions, organelles, and we're learning a lot about, how the cell works every day. We learned a lot already, but we're still learning a lot more. And one of the biggest questions that we still don't know the answer to is, how this cell came to be in the first place. What construct, what type of structure existed that eventually led to the cell as we know it today? It's, possible that a more primitive version of the cell existed before. So something like a primitive compartment, maybe something simple that had some type of boundary, maybe encapsulated some components like genetic components such as nucleic acids or similar, catalytic components such as simple peptides. We're not sure, and this is what we're actually very interested in studying. And so one of the ways to approach the origins of life research field is to find a way to, find ways where how chemicals that existed on early Earth eventually were able to assemble into this construct of the primitive cell, or some people like to say protocell, and then how this protocell evolved into modern cells. And one way to actually view this, or how to approach this question, is to approach it from both sides. We have, some information about prebiotic chemistry, what chemicals existed on early Earth. We have a lot of information about what biology looks like, what modern cell, is composed of. So perhaps one way to view how this protocell existed or what it might have looked like is to approach it from what we know, what information we know. And so let's actually kind of start from the beginning here, from the modern cell, the modern side. And one of the things that is very crucial to understand is compartments are really crucial to life. So specifically, compartments like the cell or organelles are really crucial to life. These physical constructs, they separate reactions, they promote reactions, they form barriers between different types of reactions. And so a compartment like the cell or organelles within the cell is a very important aspect that we believe a primitive, protocell has to take many of these similar functions. One type of compartment that is actually very present in biology are these, membraneless organelles, membraneless structures within the cell that they're formed through a process called liquid-liquid phase separation, and they allow, they're membraneless, so they're not bound by membrane boundaries, and also they're able to, dynamically assemble and disassemble depending on the conditions within the cell. So for example, stress granules, P granules, a lot of these types of membraneless organelles have, a lot of research has been done on them recently. And in general, there are generally two types of phase separation that we consider from a protocell standpoint, non-associative and associative. I think most of the audience here is probably more familiar with so let's say the associative version is the ones where you have two, for example, oppositely charged polymers that bind together very strongly, and they create this condensed phase outside of this dilute phase. And so these traditionally have been called coacervates, and coacervates actually exhibit a number of functions that are very relevant to a primitive system. For example, we've found that, or researchers in the field have found that coacervates can segregate and concentrate biomolecules, sometimes up to orders of magnitude. Coacervates can grow and divide. For example, this demonstration showed that electric fields can help to divide these growing coacervates into smaller parts. And coacervates also have the ability to form these more complex physical structures. For example, some of these coacervates can have multiple phases or multiple subphases. Coacervates can also scaffold more higher ordered structures like lipid bilayers. So here researchers showed that a coacervate droplet can actually, you can build this lipid bilayer around this coacervate droplet, which is maybe more approaching what a primitive cell might look like. For example, a condensed phase interior like the cytosol, and this lipid boundary on the outside like the cell membrane. So with this in mind, we actually had an idea. So one of our collaborators, originally in France, now in Italy, was an expert in these other types of self-assemblies called, DNA liquid crystals. I'm sure everyone here, a lot of people here are very familiar with liquid crystals, and we thought, could coacervates actually help to scaffold the assembly of these liquid crystals? And actually, what was very nice about the system that our collaborator Tommaso Fraccia, the system that he works with, is that they're actually built from DNA. So he has a system where you have these short DNA 12-mers. They can be shorter, they can be a little bit longer. And the DNA 12-mers, they form these nice short blunt end rods that actually stack together depending on the concentration of the DNA. And one of the components of a coacervate could be DNA. And you can actually, in general, have high concentrations of DNA mixed with high concentrations of a cationic peptide, and you might be able to form these droplets. So we decided, okay, let's try to use this system, add some cationic peptides. We used polylysine at very high concentrations, and see if we can find droplets that can help to assemble these nice liquid crystal structures. And long story short, we were able to find conditions where we have this nice droplet, and it exhibits the supramolecular chirality that is usually, shown by these liquid crystal structures. And so we found this, but it was actually a very long process because this type of structure could only exist at a specific salinity. So at very low salinities, when we had this DNA and the peptides, interacting, they formed these aggregate-like structures or precipitate-like structures. But once we started increasing the salt concentration of sodium chloride to very high concentrations, like 900 millimolar, 1 molar, you started to see the disappearance of this liquid crystal type of structure, and you get more of these isotropic droplets or even no droplets at all. And we actually thought that it might be because the sodium chloride is actually modulating the strength of the interaction between the DNA and the peptides themselves. So now we know that in general, when you find these liquid crystals, that they're often formed from this blunt end stacking, the stacking of these rods on their blunt ends. And we thought, how can we show that this is what's happening inside the droplets as well? So we actually modified the DNA a little bit, added two T-base overhang on each side, which presumably prevents them from actually stacking together. And what we found is that we couldn't reconstitute this liquid crystal character at all. We only ever found these isotropic droplets with no structure whatsoever, or no supramolecular chiral structure whatsoever. So we think that actually inside the droplets, these DNA are also actually stacking together as they would in bulk solution. A few other interesting observations that we made was that upon dehydration, let's say we started with this, this mixture of DNA and peptides at a fairly low concentration of sodium chloride. Upon dehydration, we can actually observe that the phases that it goes through, it goes through all of the phases that we observed when we scanned through the row here with increasing salt concentration. And we believe that this might be actually due to the fact that the dehydration process itself is slowly increasing the salt concentration so that these structures can exist at their various, exist in their various forms at these concentrations. And specifically, dehydration is very of interest to origins of life researchers because, on early Earth, there's not a lot of ways to generate energy to, push reactions towards completion. Dehydration is actually one of these ways where you can assume that, for example, higher temperatures, sunlight might actually result in removal of water through evaporation, and that pushes certain reactions that lose water forward. So we're really interested in general in our field to look at these dehydration type reactions. And another type of interaction, or another type of process we're interested in is temperature cycling. So again, we started here with a system which was fairly low in salt, so about 600 millimolar sodium chloride, and this exhibits this typical precipitate-like structure. And we started at 20 degrees. We slowly increased the temperature, and we found that eventually at a specific temperature, the structure changed, and it changed into this, from this precipitate-like structure into droplet-like structure, which exhibits some supramolecular chirality, possibly indicative of liquid crystal formation. And then at even higher temperatures, we found again the liquid crystal like structure may have disappeared. We then lower the temperature, and we see that on the way down, we regain some of this supramolecularly chiral structure, and at the same time, we observe a few other types of liquid crystals, not just the cholesteric type, but also columnar types. And after we did this temperature cycle, went up and then went down, we ended up finding that at 10 degrees, we still have these liquid crystal-like structures within the droplets. And this is very different from what we started with. Again, we started with this mess of precipitate-like or aggregate-like structure, and just through a simple temperature cycle, we can find the appearance of these possibly liquid crystal containing droplets. And we think that it might be because the liquid crystal structure within the droplets is actually the thermodynamic minimum of the system, and it needs this annealing step, it needs this temperature cycling step in order to find this minimum, whereas if you just mix these molecules, it can't really find that minimum. And we actually did this process at all different salt concentrations, and you can see that in some cases we're able to find some of these more columnar structures at lower salt concentrations. And we even found that the concentration of the DNA within these droplets can actually increase. So one of the functions of coacervates that is very interesting for prebiotic chemists and origins of life researchers is their ability to up-concentrate components. And in this case, we found that in some of these droplets that the DNA, we started around 1 millimolar. The concentration inside was, up to a couple orders of magnitude higher. So we think that it's really interesting. This type of coacervate system is possibly a good way to not only, introduce some type of, ordered structure into a primitive system, but also to up-concentrate some of the components within the droplets. And just as a thought here, so we think this is what's roughly happening. So going from left to right, increasing in heat and salinity, at first at lower heat or lower temperatures or lower salinity, we have very tightly packed rods indicative of this more columnar structure. And then with the peptides wound somehow binding with these DNA rods, forming these droplets. As we increase the heat or the salinity, slowly these rods start to come apart. So you see still droplets, but different type of liquid crystal structure. And then at even higher salinities and concentrations here, the rods start to disassemble from their blunt ends. And so the liquid crystal character appears to disappear, but you still have the droplets, which are produced from the peptides interacting with the DNA. And finally, at really high temperatures, at really high salinities, everything comes apart. You really can't, there's no more possibility for these peptides to interact with the DNA, and you end up with a uniform phase. So, we published some of these results previously. If you're really interested in the details, I'd encourage you to take a look at some of these papers. And specifically, Tommaso, our collaborator, he's been continuing some work on the system. For example, their recent studies, they found the ability for oligonucleotides to ligation of small, short nucleotides within these droplets without enzymatic help. They found the ability for wet-dry cycling to also introduce some more of these structures within the droplets. And recently, they've found a system which allows the polymerization of these cyclic nucleotides. These cyclic nucleotides within these liquid crystal-like structures as well. And a few years ago, we were invited to write this commentary about how possibly liquid crystals could be relevant to the origins of life, and this is published in Emerging Topics in Life Sciences. We had the opportunity to work with Noel Clark and Tommaso Bellini, who we've heard their names thrown out. And so if you're interested, I really encourage you to read this short perspective. So with the remaining part of this talk, I'd like to focus on the other direction. So we talked a little bit about how we can take lessons from the modern cell and membraneless compartments and learn more about the protocell. How about from prebiotic chemistry, from the chemicals available on early Earth? And so specifically, there's many ways that we know that molecules could have formed on early Earth. For example, through meteorite delivery, through spark discharge, like Miller-Urey style syntheses, through UV irradiation. There's many ways. And at the same time, you can actually envision that there's actually a lot of molecules that had nothing to do with biology produced at the same time. For example, when we analyzed the components of this meteorite that may have been indicative of what chemicals existed on early Earth, you can find a number of different chemicals and a number of different chemical formulas exhibited. When you analyze the reaction products of Miller-Urey reactions, you find a number of different products, and there's no way that all of these products are biomolecules or directly relevant to biology. And so rather than thinking about how biomolecules formed on Earth, we're interested in thinking about, what molecules were available on early Earth that may either not have been present in, are either not present in modern biology or have a different role than they have in modern biology. And whether these non-biomolecules, as we call them, can catalyze life's emergence. And specifically, there's a system that we like to use. This is a standard system of alpha hydroxy acids and polyesters. So alpha hydroxy acids, the top row, they're very similar in structure to amino acids, with the amino group being replaced by a hydroxyl group. And although hydroxy acids are present in biology, for example, lactate is in metabolism. You can find other types of hydroxy acids in various cell membranes or cell membrane components of different bacteria. They're not very much viewed as essential biomolecules. However, why we like to use this system as a model system is that you can actually take monomers of hydroxy acids, subject them to dehydration, and they lose water, and they very readily form polymers. And these are polyester polymers. This is shown in B. There we go, in B. And this is a very, a very thermodynamically, this reaction happens very readily due to thermodynamics. I think the delta G is around 0. So even just simple dehydration will form lots of these polymers. And so what we found is that if you started with a number of these, each of these monomers, if you subject each of them to simple dehydration, you actually form this slew of polymers in the reaction product. So starting with, for example, lactate monomers, if you subject it to dehydration synthesis through heat at just 80 degrees for about 1 week, you end up with product polymers that are polydispersed, but up to around 40 or maybe even 50 units long. And at the same time, you find that they can form these gels. So these gels are, are exhibited in almost all of the formulations that we worked with of these 5 different hydroxy acids we tested. And if you rehydrate these gels, you actually find droplets. So these droplets appear to be membraneless. They're certainly made from the polyester products that we generated. And these droplets are interesting because, for example, they can segregate dyes, might be very important for a primitive system to be able to segregate molecules. They can also coalesce. So here's a droplet system with a little bit of salt added. And so you can see over time that the droplets can actually coalesce and recombine, or combine, if you will. And they can also segregate RNA, which has been proposed as one of the molecules essential for the origin of life. And the RNA can still function. For example, here we found that a ribozyme, a functional RNA, an enzymatic RNA, is still functional in the presence of the droplets. So we proposed that these droplets, these polyester droplets, are relevant to the origins of life as protocells, as possible primitive cells before the emergence of the membrane, for example. And so one of the things that we're interested in is, of course, we're using these hydroxy acids. We're interested in whether the differences in monomer chirality actually can affect the any structural properties of the products that are formed. So long story short, first of all, we did some basic studies, and we found that for lactic acid and for phenolactic acid, the products, the synthesis procedure, the synthesis products, it didn't matter which chirality of monomer we used, whether it was L, D, or DL. We also found that for the same system, that the structure of the droplets, it really wasn't affected at all by whether we use DL or DL. So we published this in a small paper, and we thought that this is only true for LA and PA. How about other systems? So earlier you heard this nano talk by Chen, and so he joined the lab in 2021, and first he worked on a study where he found that these droplets really like to uptake salts in specific ways. And the next question he thought about is, are there other types of systems, other types of hydroxy acids that we can test where maybe we find a difference whether they polymerize or not based on their chirality. And so in a very interesting stroke of luck, we were working with tartaric acid. And before I go any further, this data has just been accepted into a journal. Please don't take photos. You can read the paper when it's out soon. And so we were studying tartaric acids, and so we were subjecting tartaric acid monomers to dehydration synthesis, and we found that, okay, well, they don't really seem to polymerize. And then our collaborator, Ray-Chin, he was also studying tartaric acid monomers, and he was like, oh, well, we found these tartaric acids, they really like to polymerize. And so we're like, what's going on? We're using the same thing, right? Turns out we weren't. So what Ray-Chin was using, he was working with D tartaric acids or L tartaric acids, enantiopure tartaric acids, and we're working with DL, so racemic. And so we actually found that for whatever reason, which may become clear later, tartrates or tartaric acid only appears to polymerize readily through dehydration synthesis in an enantiopure form, whereas if they're racemic, they don't really polymerize. And then in another stroke of luck, we started, because Chen was working with salts, we started playing around with different salts, and we actually found that if we added calcium, that the polymerization products kind of equalized between racemic and enantiopure tartrates. We weren't totally sure why, and so we did a little bit more digging, and we found that. So these are conversion yield. These are the amount of monomers converted into polymers as measured by NMR, and we found that in the presence of high concentrations of calcium, that the yield was roughly similar between all different chiralities or chiral ratios. Whereas for low concentrations of calcium or no calcium, racemic mixtures of tartaric acid really didn't seem to like to polymerize. Why? So first we thought, oh, maybe it's the number of D and number of L types of molecules in the mixture might be directing this effect. And we did this study with mesotartaric acid, where in principle the number of these types of D or L linkages would have been the same as racemic tartaric acid, and actually in the presence of calcium, this also doesn't, in the presence of calcium, it doesn't seem to polymerize, and without calcium, it seemed to polymerize. So we thought, okay, it's probably not due to the total numbers of these, types of linkages. And instead, we found that doing some further studies, we think that it's due to the fact that the racemic TA, the racemic tartaric acid, may be in some type of geometric conflict with each other where it's not able to polymerize due to geometries, whereas the enantiopure tartrates may be able to polymerize. And of course, we did some further studies, and we also found that this process is very unique in that only in the presence of calcium we see this equalization of the polymerization rate. With magnesium, with sodium, there's almost no effect, and racemic tartaric acid really can't polymerize, whereas in the presence of calcium, racemic tartaric acid seems to be able to polymerize. And it's possible that this binding of the calcium with tartaric acid, it allows it to achieve a specific type of geometry or a type of formation that allows this polymerization to occur, whereas in the absence of the calcium, this wouldn't have been possible. This leads us to speculate that, for example, lower calcium environments, for example, some freshwater systems might actually promote polymerization of these pure enantiopure polymers, enantiopure polymers, possibly leading to more homochiral polymers, whereas with high calcium environments, perhaps polymerization, it leads to polymerization of atactic polymers, and you get less of these homochiral polymers. Some of the other things we learned was that, as you know, calcium tartrate forms crystals, and these crystals are really apparent at when we have a lot of racemic tartaric acid, but with enantiopure tartaric acid, they don't really exist. And it may be because calcium tartrate with racemic tartrate has a much lower solubility than some of the calcium tartrates with more enantiopure tartrates. And we also found that within these crystals, it's very likely that the crystal itself contains roughly equal amounts of the D and the L tartrate. And finally, we also found that there are systems where we could find droplets composed of polyesters, crystals composed of hydroxy acids that kind of simultaneously existed. So we're thinking about ways by which, or where a primitive system could have been able to use, for example, the droplets as some type of microreaction vessel, and these tartrate crystals as stores from where it can draw more monomers for reactions. So again, paper is accepted. It'll be published in a few weeks. Stay tuned, and please visit Chen's poster, which has some more details. And if you're interested in learning more about the polyester system, please feel free to look up some of our other studies. And finally, I'd like to thank all of our funding sources. None of this can be possible without funding, so I need to thank all of our funding sources. Our collaborators, Tommaso, Chen, and Rachin. Chen and Rachin are here. Again, please go meet them and talk to them. And all of our other collaborators, our colleagues from LC, from former Tokyo Tech, and around the world. A little bit about our institute. So LC, the Earth Life Science Institute, it's an interdisciplinary institute that's set up to study the origin of planets, Earth, and life on Earth. And we're in Tokyo, in Meguro. So please come visit if you ever have a chance. And we recently started a graduate program. It's a 5-year graduate program that encompasses both master's and PhD parts, which is, I think, very unique in Japan, which is often the master's and PhD are separated. And every year, we actually will fully fund 10 students, including a stipend. And specifically, they're encouraged to work with multiple faculty from the institute to have more exposure to different ideas or different disciplines. And so if you're interested, or if you have students, undergraduate students who are interested, there will be another application deadline at the end of the year for spring 2026 enrollment. I think this is a really great place for students to go and study different topics related to the origin of life. So thank you very much for your time. I'm happy to take questions.